Welcome back to the deep dive. Today we are undertaking a really critical mission. We're cracking open the foundational language of the central nervous system.
We really are.
This is the chapter chemical neurotransmission that is basically the roadmap for understanding how every single psychiatric drug and, frankly, every neurological disease impacts the brain.
It's the key.
So if you want a genuine shortcut to being well-informed on this topic, you really have to know this molecular story, inside and out.
And our mission today isn't just to, summarize it. It's to translate it. We're going to take this incredibly complex, step-by-step molecular cascade of how neurons talk to each other and just break it down.
So you can actually visualize it just through audio.
Exactly. We'll start with the physical layout, then the modes of communication, and then we'll get into the deep chemical consequences that, they change the very nature of the neuron that receives the message.
Okay, let's start with the hard boundaries then. Neurotransmission, it fundamentally rests on two parallel systems. First, there's the anatomical basis. People call it the hardwired system.
Right. Think of it like old school telephone wires. The physical infrastructure, every neuron is a wire. It's got the soma, which is the command center, the cell body.
And the dendrites for receiving signals.
The dendrites for receiving, and then that long axon that sends the signal out, which ends in the presynaptic terminals. It's a very clear structure.
And these connection points, the synapses, they can be different types. Axon-dendritic is common, axon to dendrite.
Axosomatic, axon to the cell body, sometimes even axon to axon. But the key rule anatomically is that the information flows one way, anterograde. It's a one-way street.
But that's just the wiring. The real story for modern medicine, and for us today, is the chemical basis, the chemically addressed nervous system.
This is where psychopharmacology lives and breathes. It's all about the signal itself, not just the wire.
And before we go any further, we have to name the main characters. The 6 key neurotransmitters that are targeted by, well, pretty much every psychotropic drug out there.
They are serotonin, norepinephrine, dopamine, acetylcholine, glutamate, and GABA. When you hear about an antidepressant, an antipsychotic, anything, it's almost certainly messing with one or more of these 6 chemicals. They're the language we're trying to decode.
So let's move from that structure to the function. Segment 1, the 3 modes of neurotransmission. The classic model, the one we all learned, says the message is electrical inside the neuron, but it's chemical between neurons.
That's classic neurotransmission. An electrical pulse hits the end of the line, the terminal, and forces this chemical payload across a tiny gap, the synaptic cleft, to hit a receptor on the next neuron.
A very precise point-to-point delivery.
Incredibly precise. And we're talking about trillions of these connections in the brain. It's staggering.
But that's the classic model.
Yeah.
The really fascinating part is the exceptions, the twists that make the system so much more dynamic. First up is retrograde neurotransmission.
All right.
This idea that the neuron receiving the signal can actually talk back to the one that sent it.
It's a sophisticated regulatory loop. It's the receiving neuron sending a message back to the sender, often saying something like, okay, ease up a bit, or let's change our connection long term.
So can you give us a concrete example of that kind of talk back?
Oh, absolutely. The source highlights endocannabinoids, ECs. Some people call them endogenous marijuana. They're synthesized on demand, postsynaptically so, after the message is received.
Yeah.
And then they just diffuse backward across that gap and hit CB1 receptors on the presynaptic terminal. Functionally, it just dampens the reverse of the next signal. It's an immediate feedback system.
The receiver says, got it, now hold your fire for a second.
Exactly. And then there's a longer-term example with neurotrophic factors like NGF, nerve growth factor. These are released by the postsynaptic cell, taken up by the original neuron, and then actively transported.
All the way back up the axon.
All the way back to the presynaptic cell's nucleus to interact directly with its genome. That is a message traveling a huge distance to make a long-term fundamental change.
That's wild. Okay, so the second twist is volume neurotransmission, non-synaptic diffusion. This seems to completely contradict that whole hardwired idea.
It does. It suggests the brain isn't just a network of precise wires, but it's also, a kind of chemical soup.
Yeah.
It's the shift from landlines to cell phones. You don't need a direct wire. You just need to be in the transmitting radius.
So the neurotransmitter can spill over from its synapse and just drift over to receptors that are far away.
And hit receptors that have no physical synaptic connection to the neuron that released it. It's a broadcast message, not a point-to-point call.
And the practical relevance of this is huge, especially when you look at dopamine. The source compares the striatum with the prefrontal cortex, the PFC.
Right. In the striatum, which is all about motor control, you need dopamine to be incredibly precise. So nature puts a ton of DATs there. dopamine reuptake pumps that just vacuum up the dopamine instantly.
So it keeps the signal clean and local.
Exactly. But in the PFC, which handles planning and cognition, you want that volume transmission. So there are very few DETs there. The dopamine is free to diffuse, to float around and modulate a whole neighborhood of neurons. It's crucial for coordinating complex thought.
Okay, let's move on. Segment 2, excitation, secretion, and signal transduction. We've got the chemical messenger. How does the electrical impulse actually become that chemical messenger? This is the critical moment, right? Excitation, secretion, coupling.
This is the conversion point. When the electrical impulse zips down the axon, reaches the terminal, it first pries open voltage-sensitive sodium channels. That keeps the signal going. But then, crucially, it opens voltage-sensitive calcium channels.
And calcium rushing in, that's the trigger.
That is the trigger. Think of calcium K++A as the key that unlocks the floodgate. It rushes into the terminal, and that influx causes these little bubbles full of neurotransmitters, the synaptic vesicles, to fuse with the membrane and spill their guts into the synapse.
And just like that, an electrical event becomes a chemical one. But once that chemical hits the other side, the journey is really just starting. Now we follow the message inside the cell. The signal transduction cascades. The source calls this the molecular pony express.
Which is a perfect description. The message isn't just received, it's handed off. Sequentially, from the first messenger, the neurotransmitter, to a second, then a third, a fourth, all these molecular riders, until it hits the ultimate destination, the genome.
And this is where the time course becomes so important for treatment. That initial binding, the second messenger being formed, that's milliseconds. But the real consequences, new proteins, changes in gene expression, that takes hours to days.
That lag is everything. It explains why most psychotropic drugs, which kick off these cascades, take weeks to show their full clinical effect. You're waiting for the cell's entire manufacturing process to change in response to that initial signal.
And inside the cell, there's this constant battle happening. A molecular conflict between two families of third messengers.
Oh yeah, it's a constant push and pull between kinases and phosphatases. Kinases are the activators. They add phosphate groups to other proteins. It's called phosphorylation, which usually turns them on.
And the phosphatases?
They're the immediate counterforce. They rip those phosphate groups right back off, reversing what the kinase just did. They're the molecular reset button. So the ultimate biological response of that cell is determined entirely by the balance of power between these two.
Which brings us to segment 3, the 4 major cascade systems. We'll focus on the 2 main ones that drugs target, the G-protein link system and the ion channel link system.
Let's start with the G-protein linked system. This one uses a chemical second messenger, like CAMP. You need 4 parts to come together to make this machine work.
Okay, so first, the first messenger NT binds to the receptor. A 7-transmembrane receptor, meaning it weaves through the cell membrane 7 times.
And that binding is like the key turning in the lock. It changes the receptor shape. That new shape allows the third piece, the G protein, to bind to it, which in turn changes the G protein's shape.
It's a molecular relay race, where each handoff is a physical change.
It is. And that newly shaped G protein then detaches, slides over, and locks onto the fourth piece, an enzyme like adenylyl cyclase. That activates the enzyme, which then starts churning out the second messenger, CMP. It's this beautiful domino effect.
And what does STAMP do?
It finds its target, an inactive protein kinase dimer. You can picture this kinase as a weapon with a safety on. The TMP binds to the regulatory units, the safety, and causes them to float away. Now the kinase is active, unleashed, ready to go shoot phosphate groups onto other targets.
Okay, so that's the complex chemical relay. Now contrast that with the ion channel linked system.
This one is simpler, at least at the start. The neurotransmitter binds, a channel opens, and an ion, usually calcium K++, rushes in. The calcium itself is the second messenger, no middleman needed.
But once it's inside, calcium isn't so simple. It has a dual role, doesn't it?
Yes, and that makes it incredibly efficient. That influx of calcium can immediately turn on a kinase, like CAMK, to start adding phosphates. Or it can turn on a phosphatase like calcineurin, which is ready to rip phosphates off.
So the same signal, calcium, can drive the cell toward activation or deactivation.
It all depends on the machinery it finds once it gets inside.
And all these signals from both systems, they eventually hit the fourth messenger, phosphoproteins.
These are the workhorses of the cell. The ion channels, the pumps, the enzymes, they're just sitting there waiting. A kinase adds a phosphate, and their function changes instantly. A phosphatase removes it, and it changes back. This is how the immediate excitability of the cell is managed.
So we've gone from the synapse through the cytoplasm, now for the long game. Segment 4, the path to gene expression and epigenetics. This is the most powerful goal of all.
Right. For any change to be truly lasting, the neuron has to change the proteins it makes. And to do that, you have to talk to the genome. The activated kinases from both systems we just discussed can actually travel into the cell nucleus.
And once they're in there, they phosphorylate the ultimate decision maker, a transcription factor called CREB.
Activating CREB is like sounding the alarm. It's the signal to turn genes on. And when a gene is turned on, it's like deploying an army, and it happens in stages.
The first wave of troops being the immediate early genes like CFOs and KIJUN.
These are your special ops. They respond incredibly fast, within about 15 minutes. The proteins they produce, FOs and JUN, are the fifth messengers. They team up to form an even more powerful transcription factor, the sixth messenger, often called a leucine zipper.
And that leucine zipper then goes on to activate the much larger army of late genes.
Exactly. This two-step process allows for massive, coordinated, and yet delayed changes. And these late genes are what produce the really profound effects. Synaptogenesis, building new connections, making new receptors, all the things that underlie learning, memory, and sustained recovery from illness.
And running parallel to all of this is epigenetics. This is a concept that, it really ties everything together: environment, stress, drugs.
It's the storyteller. If the genome is the lexicon, all the words a cell could possibly use, the epigenome is the story. It dictates which words, which genes are actually spoken and which are silenced.
And it does this by changing the physical structure of chromatin, right? The DNA wrapped around those histone proteins.
Think of it like tightly wound spools of yarn. To silence a gene, you tighten the winding. You do this through methylation and deacetylation. This compacts the chromatin so much that the machinery can't even get in to read the gene. The molecular gate is closed.
So to activate it, you do the opposite. You loosen the winding.
Demethylation and acetylation. It decompresses the chromatin, exposes the gene, and allows it to be transcribed. And here's the kicker. This pattern isn't fixed. It changes in mature neurons based on your experiences. Chronic stress, drugs, even psychotherapy can change the epigenetic state of your brain.
That's incredible. Finally, let's touch on segment 5, a brief word about RNA. We think of mRNA as the blueprint, but it's more complex than that.
Much more. First, you have alternative splicing. Imagine the initial RNA transcript is like raw footage. You can edit that footage in different ways to create multiple distinct movies from the same raw material. One gene can produce many different proteins. It's a huge source of molecular diversity.
And then there's RNA interference, or RNAi. This is where RNA actually works to block protein synthesis.
It's a quality control system. These little non-coding RNAs, like miRNA, are processed by an enzyme called dicer. They then get loaded into a complex called RISC, which acts like a search and destroy missile. It finds a specific mRNA blueprint and prevents it from being translated into a protein. It's a crucial off switch.
So we started with a simple electrical pulse, and we follow this incredible chain reaction that can end up rewriting the genetic instructions of a neuron for life.
That is the ultimate conclusion, isn't it? Chemical neurotransmission is, at its core, a conversation between genomes. The presynaptic genome sends a chemical message that ultimately tells the postsynaptic genome how to change.
We said that today, most drugs target those very first steps, the neurotransmitters, the receptors. But this whole conversation suggests the future is about targeting those deeper, long-lasting molecular cascades.
The cascades, the epigenetic mechanisms, exactly. And that incredible transformation where a fleeting signal causes a lasting structural change, that leads us to our final thought for you.
Okay.
If a brief puff of chemical neurotransmission can trigger a profound postsynaptic reaction that takes hours to develop and can last for days, weeks, or even a lifetime, what complex behaviors might be driven by changes in gene expression that were originally triggered hours or even days ago?
A compelling idea to consider. Thank you, as always, for engaging in this deep dive with us.